You know, inside you, right this very second, there's an invisible war being waged. Seriously, it's this high-stakes microscopic drama where your body is constantly deploying its elite first responders to protect you. Today, we're going to pull back the curtain on this incredible process.
Think about it. It seems so minor, doesn't it? A tiny little paper cut. But that small break in your skin actually triggers a massive alarm, launching one of the most sophisticated emergency response protocols you can imagine.
So how does this whole rescue mission even unfold? Well, you can think of it like a drama in 5 acts. What you're seeing here is the entire journey of a single white blood cell, a neutrophil, as it goes from the superhighway of your bloodstream to a battlefield out in your tissues. Let's break this down, step by heroic step.
Okay, so when bacteria get in or your tissue gets damaged, a chemical SOS signal just floods the area. And who answers the call? The elite special forces of your immune system, a type of white blood cell called the neutrophil. They are always the first on the scene, and their job is pretty simple: get to the problem and neutralize the threat.
But here's the thing. Just getting to the fight is a huge part of the battle. Our hero, the neutrophil, is currently cruising along inside a blood vessel, which is like a biological superhighway. To get to that infection, it has to pull off a daring escape right through the wall of that vessel.
And this is not just some simple hop. Oh no, it is a highly orchestrated 4-step process. Think of it like a cellular obstacle course. We've got margination, then rolling, then adhesion, and for the grand finale, diapedesis. First up is margination. See, normally blood is flowing pretty fast, but near an injury, inflammation causes everything to slow way down. It's basically a biological traffic jam. And this slowdown gives our neutrophil the chance to get out of the fast lane and just kind of drift over to the edge, or the margin, of the blood vessel wall. So how does the body slow our little hero down even more? Well, the cells lining the vessel wall start to sprout these sticky proteins called selectins. You can think of them as molecular speed bumps. The neutrophil has its own parts that briefly grab onto these selectins, which makes it catch and release, just tumbling slowly along the vessel wall, kind of like a ball rolling to a stop. Rolling is great, but we need the neutrophil to stop at the exact right spot. So chemical signals activate another set of proteins on the neutrophil called integrins. And these aren't speed bumps, these are more like superglue. They lock on tight to the vessel wall, bringing our rolling hero to a complete and total stop. Okay, so our neutrophil is stuck to the wall, but it's still inside the blood vessel. How does it get out? This is the last step, diapedesis. And it's truly incredible. The neutrophil uses a special molecule, PCAM1, almost like a key to unlock the little gaps between the cells of the vessel wall. Then it totally changes its shape, gets all long and thin, and literally squeezes itself through that tiny gap and out into the tissue. Wild, right?
And just like that, the great escape is complete. Our hero is now in the tissue. But the mission is far from over. Now it has to find the bad guys.
So, how does it know where to go? It basically follows a scent, a chemical breadcrumb trail left by the bacteria and by your own damaged cells. This process is called chemotaxis. All these chemicals you see here, like IL-8, NC5A, create a gradient, a trail, and the neutrophil just follows it straight to the source of the problem. This brings us to the climax of the fight, phagocytosis. The neutrophil tracks down the enemy, extends its own cell membrane, these things are called pseudopods, and completely wraps itself around the bacteria, pulling it inside into a little prison. But trapping the enemy isn't enough, right? It has to be destroyed. And to do this, the neutrophil doesn't just pull out a weapon it has lying around. No, it manufactures one of the most powerful chemical weapons in all of biology, right there on the spot. This whole thing is called the oxidative burst, and it's just an amazing piece of natural engineering. In a super-fast 3-step reaction, an enzyme called NADPH oxidase turns oxygen into a toxic compound called superoxide. Then that gets turned into hydrogen peroxide, and finally, in the last lethal step, another enzyme converts the hydrogen peroxide into hypochlorous acid, which you might know it as the active ingredient in household bleach. Yes, you heard that right. Your cells literally create bleach to kill germs. This system is just breathtakingly effective. But because it's so complex, sometimes things can go wrong. So what happens when these cellular heroes can't actually fight? Remember that integrin superglue we talked about, the stuff that stops the neutrophil on the vessel wall? Well, in a rare genetic condition called leukocyte adhesion deficiency, or LAD, that glue is basically defective.
The neutrophil can slow down and roll, but it can never get that firm grip. So it's essentially trapped in the bloodstream, unable to get out and fight. And this leads to really severe recurrent bacterial infections.
Now, what if the neutrophil can get to the fight, but its weapon system is busted? That's what happens in a condition called chronic granulomatous disease, or CGD. Here, the problem is with that NADPH oxidase enzyme, the very first step in the bleach-making process. The neutrophil can still eat bacteria, but it can't kill them very well, which leads to all sorts of chronic inflammation and serious infections. So, we've seen the mission and what happens when it fails, but when everything goes right and the infection is cleared, what happens to our hero? Before we get to its final fate, let's do a quick mission debrief. This table is a great little summary. Remember the speed bumps for rolling? That was the selectins. The superglue for adhesion? That's our friends the integrants. The key for squeezing through the wall is PCAM1, and that breadcrumb trail it follows is made of signals like IL-8. These are the molecules that make the whole incredible mission possible.
So after all that, after the battle is won, our hero doesn't get a parade. Its job is done. These cells are built for a short, intense life. Within about 24 hours, they go through this clean, programmed self-destruction called apoptosis. They sacrifice themselves to prevent any extra unnecessary inflammation. It's really the final, heroic act in a very short but very vital life. So next time you get a paper cut, just stop for a second and appreciate the incredible microscopic drama that's unfolding right under your skin. It's an elegant, violent, life-saving battle fought by billions of these cellular heroes just to keep you safe. This invisible war happens all the time. Doesn't that just make you marvel at the extraordinary machine you get to live in?